Welcome to Section 8 of Immunology. In this section, we'll be discussing macrophages. Let's get started. Macrophages are very important during inflammation, and understanding the details of macrophages is easiest when you compartmentalize their activities during acute inflammation and chronic inflammation. During the acute inflammation video, Section 2 of this chapter, I explained that macrophages will infiltrate the tissue within 2 to 3 days. They will also perform phagocytosis and oxygen-independent killing. They will also perform cytokine secretion and initiate possible abscess formation. And this is the acute inflammation overview image, which we discussed in Section 2. And here we can see a macrophage in the postcapillary venule, and it will infiltrate the tissue within 2 to 3 days. It will then perform phagocytosis. It will then kill the pathogen, and then it will secrete cytokines. We've shown 3 examples here. And lastly, they can initiate possible abscess formation. Those are the activities of macrophages during acute inflammation, in a nutshell. But let's dive into the details of each of those activities to the level that you need to know for step 1, starting with phagocytosis. To perform phagocytosis properly, macrophages must identify what cells to engulf. Also, the ability to engulf or phagocytose pathogens is greatly enhanced by opsonins. Do you recall what the 2 primary opsonins are for macrophages? C3b and IgG. This slide lists the surface proteins that are relevant for macrophages during phagocytosis. So macrophages have a CD14 protein. This is a toll-like receptor, and this will bind to pathogen-associated molecular patterns, or PAMPs. The prime example of a PAMP is lipopolysaccharide, which is on gram-negative bacteria. This CD14 can also bind to DAMPs, which are damage-associated molecular patterns. A good example of this would be necrotic tissue. So this top bullet point explains how macrophages identify what to engulf, and they do this with CD14. The next 2 bullet points show how they interact with opsonins. They do this using an Fc receptor, which will bind to the Fc portion of IgG. Again, that's one of the 2 major opsonins you need to be aware of. Another receptor is the C3b receptor. This will bind to C3b, which again is an opsonin. So now we can see what surface proteins or receptors are necessary for macrophages in order to interact with opsonins. This image shows the detail of what we just discussed. In the center, we have a macrophage, and if you look at the top, on the surface, you can see a CD14 protein. Again, that's a toll-like receptor, and it will bind to PAMPs, the best example being a lipopolysaccharide, but it can also bind to DAMPs. And if we look down to the left side of the macrophage, we see the Fc receptor, and this will bind to the Fc portion of the IgG antibody. And it also has a C3b receptor, which will bind to C3b and more easily engulf the pathogen. After ingesting the pathogen, the macrophage needs to kill it, and it does this through oxygen-independent killing. Recall from the neutrophils lecture in Section 3 that there is oxidative killing, which is the primary means by which neutrophils kill phagocytosed pathogens. However, neutrophils can also use something called non-oxidative killing, or rather, oxygen-independent killing. And it's this oxygen-independent killing that macrophages mainly use. So here's how macrophages use oxygen-independent killing. Following phagocytosis, they will use lysozyme. Lysozyme hydrolyzes the bacterial peptidoglycan, which results in lysis of bacteria. Going back to this image, we see the box for phagocytosis right here. And you can see that phagocytosis results in a phagosome, the vesicle that holds the pathogen when it's engulfed. It's then combined with a lysosome. And notice that the lysosome is full of lysozyme. And these 2 vesicles will combine to form a phagolysosome. And this exposes the pathogen to lysozyme and kills it. Now, macrophages also secrete lots of cytokines during acute inflammation. This table lists the cytokines that macrophages and dendritic cells produce. In the acute inflammation lecture, Section 2, we discussed IL-1 and TNF briefly. And you can also call TNF TNF-alpha, but they're the same thing. But on this table, we have a more thorough list, which includes interleukin 6, interleukin 8, and interleukin 12. All the details on this table are very important to know for step 1. However, don't be intimidated by this, because following this video is an image mnemonic to help you really lock in and memorize all of these details. So after that video, you'll have all these details down cold. Right now, I just want you to know that macrophages secrete cytokines. Next, let's dive into the details of possible abscess formation. Abscess formation, by definition, is neutrophils and pus surrounded by a wall of fibrosis. And the process is basically macrophages stimulating fibroblasts. And that makes sense, because fibroblasts cause fibrosis. So how do macrophages cause an abscess? The macrophage stimulates the fibroblasts, which then surrounds the neutrophils and pus. And that's really all you need to know about abscesses. Here's an image showing an abscess of the skin. So again, this is pus surrounded by fibrotic tissue. We just covered the activities of macrophages during acute inflammation. Now let's discuss their roles during chronic inflammation. During chronic inflammation, think of macrophages doing three things: local antigen presentation, cytokine secretion, and possible granuloma formation. Notice that macrophages form granulomas during chronic inflammation, but they form abscesses during acute inflammation. I found that to be a helpful way to compartmentalize abscesses and granulomas from macrophages. Now let's dive into the details of local antigen presentation. After phagocytosis and oxygen-independent killing, macrophages will present antigens locally to activated T cells. This leads to re-stimulation of activated T cells. And I want to emphasize that word local. This means that macrophages don't travel to the lymph nodes and activate T cells. Instead, they stay in the inflamed tissues. Do you remember what cells actually travel to the lymph nodes? Those are the dendritic cells. So dendritic cells will activate T cells for the first time, where macrophages really just perform re-stimulation of already activated T cells. This is the chronic inflammation overview figure, which was discussed in detail in the previous video. Macrophages, which you can see up here, will present antigens to cytotoxic T cells, which you can see here, and helper T cells. And so we've written here antigen presentation, in parentheses, re-stimulation. We do this as a way to help you remember that macrophages are re-stimulating already activated T cells. Now let's discuss the relevant surface proteins in antigen presentation. The top two bullet points list the surface proteins needed for T cell re-stimulation, where this last bullet point really identifies the surface protein that benefits the macrophage when connected to the T cells. So let's start with these top two. Macrophages have MHC2 and B7. B7 is also called CD80 or CD86. And this will perform helper T cell re-stimulation. It also has MHC1 and B7, which is again CD80 or CD86, and this is for re-stimulation of the cytotoxic T cells. Now these MHCs, MHC1 and 2, are major histocompatibility complexes, and those were discussed in the previous video. Going back to this image, after phagocytosis and oxygen-independent killing, the macrophage will perform antigen presentation. And it does this by taking the parts that have been broken up from the pathogen and then presenting those proteins, those antigens, on the MHC2 molecule. Now this image demonstrates an MHC2, which will activate T helper cells. But keep in the back of your mind that if the macrophage were re-stimulating a cytotoxic T cell, this would be an MHC1. Anyways, this MHC2 will bind to a T cell receptor, and that's the first signal. So this is one of two necessary signals for T cell activation. An additional component to antigen presentation and T cell activation is this B7 up here. This will bind to CD28 on the T cell. This is the second signal. Both are necessary for T cell activation, which you can see here. And once these T cells are reactivated, the helper T cell will release cytokines. And there are multiple types of helper T cells, which will be discussed in great detail in section 11 of this chapter. But the point here, as shown in this little box, is that the cytokines the activated T helper cell releases depends on the TH subtype, which could be TH1, TH2, TH17, or Treg. Now that we've covered these top two, relevant for T cell activation, let's talk about this third one, the CD40 protein, which is important for macrophage stimulation. Once stimulated, this will allow for continued phagocytosis, oxygen-independent killing, and more antigen presentation. Going back to this image, we can see the CD40 right here on the macrophage, and we can also see CD40 ligand here on the T helper cell. And we can see that this relationship really benefits the macrophage, with an arrow pointing to macrophage activation right here. What this means is the macrophage will then be upregulated to continue cytokine release, continue phagocytosis, continue oxygen-independent killing, and continue antigen presentation. Now this is totally logical. If the actions of the T cells are needed to fight the infection, then the actions of the macrophage will also be needed. So it makes sense that this interaction with the macrophage and the T cell would also stimulate the macrophage. Now I've shown here several different cytokines that macrophages can release. But going back to this slide, I want to remind you that cytokine secretion depends on the helper T cell subtype. For example, stimulated T helper 1 cells will release interferon gamma. Interferon gamma will then stimulate the macrophage to release more interleukin 12. So again, if we were to assume that this T helper cell was a Th1 cell, it would release interferon gamma, which would then stimulate the macrophage to release more IL-12. But again, we'll help you memorize these macrophage cytokines in the following video. So as you can see, there are several surface proteins you need to be familiar with regarding macrophages. We've covered all those involved in antigen presentation, and all those involved in phagocytosis. Now, the last one to be familiar with is CCR5. This is a surface receptor that HIV needs. HIV will recognize CCR5, bind to it, and then enter the macrophage and do all the nasty virus-related stuff that HIV will do. And for more information on HIV, please see Viruses, section 14 of our microbiology chapter. And we want you to have a reference for that last detail in your text, so we've included that information here. The surface protein that's targeted by HIV is CCR5. Continuing with chronic inflammation activities, I want to point out again that they will continue secreting cytokines. So cytokine secretion is a part of chronic inflammation and acute inflammation. Lastly, let's dive into the details of possible granuloma formation. When it comes to granulomas, it's really important to think about chronic inflammation because it can be subdivided into two types, which are defined by granulomas. One type is non-granulomatous, the other type is granulomatous. And granulomas can be caseating or non-caseating. When it comes to non-caseating granulomas, it's important to know how they're formed. Here's what happens. Th1 cells will release interferon gamma. This causes the macrophages to become epithelioid cells, also called epithelioid histiocytes. Some of these epithelioid cells will combine to form multinucleated giant cells. And it's these epithelioid and giant cells that surround the infection, which is what defines a granuloma. Now let's talk about the appearance. Granulomas are basically a collection of these giant cells and epithelioid cells. And the epithelioid cells are cells with ovoid nuclei surrounded by lots of pink cytoplasm. I'll show you what this looks like. Next, granulomas are surrounded by lymphocytes. And in chronic inflammation, what lymphocytes generally invade the tissue? That would be T cells. But in granulomas, we're getting even more specific. We're thinking specifically of Th1 cells. And that makes sense that we'd be dealing with T helper 1 cells. After all, it's these Th1 cells that release the interferon gamma, causing the granuloma to form in the first place. This is a histological image showing a biopsy of a non-caseating granuloma. In this circular area, we can see the granuloma here, and we can see lots of epithelioid cells. Again, those are also called epithelioid histiocytes. Notice the oval nuclei, and each one of these cells surrounded by lots of pink cytoplasm. And that's why there's so much pink in this granuloma. It's that pink cytoplasm from the epithelioid cells. On the periphery, out here, we can see lots of lymphocytes. And again, these would be T cells. And this particular granuloma doesn't have any obvious giant cells. So let's do another example. We can see the granuloma and lots of epithelioid cells with that abundant pink cytoplasm. And we can see all the surrounding Th1 cells. And in this image, we can actually see a giant cell, which again is just a bunch of epithelioid cells that have merged together. So we see one cell with a lot of nuclei. Next, let's discuss caseating granulomas. Now, as far as formation, they are formed in the exact same way as non-caseating granulomas. And how do those form? Well, it's Th1 cells that release interferon gamma. Those will stimulate the macrophages to become epithelioid cells and giant cells. And the appearance of caseating granulomas is also the same as non-caseating granulomas, but there's also caseation. So what is caseation? Well, this is basically central necrosis. And all those cells that are necrotic will lack nuclei. And when you see this central necrosis in your granuloma, you think, okay, this is caseating, which means we're thinking of a TB infection or a systemic mycosis. And systemic mycoses include histoplasmosis, blastomycosis, coccidiomycosis, and paracoccidiomycosis. And if you're worried about TB or the systemic mycoses, just go see our microbiology section, which will help you memorize all of these details through image mnemonics. Here's a histological image of a caseating granuloma. We can see the collection of Th1 cells out here, and we can see the epithelioid cells. We can also see a giant cell, and in the center of this big granuloma, we can see this big area of central necrosis. And as you can see, all the material in this area totally lack nuclei. So this is a caseating granuloma. So what infection would you be thinking of? That would be TB or systemic mycoses, which are fungal infections. And there's one last detail you need to know about granulomas, and that is how they're maintained. Here's what happens. Epithelioid cells, again, those are formerly macrophages, will secrete TNF, which again you can call TNF-alpha. And this causes the granuloma to remain intact. Now, TNF inhibitors, or TNF-alpha inhibitors, will cause granuloma breakdown, because after all, that granuloma needed that TNF in order to stay intact. So if it's being inhibited by a drug, then the granuloma will break down. And this is a very important thing to remember because this means that there can be possible disease dissemination. For example, disseminated TB. So this is why whenever a patient is given TNF inhibitors, they need to be screened for TB because if they have dormant or latent TB inside their body with all those organisms surrounded by granulomas, then if you give them TNF inhibitors, it'll break those down and you can make their disease super bad because they can get this disseminated TB. Now that we've covered the details that you need to know about macrophages, let's do a question to apply what you've learned. Within tissue experiencing chronic inflammation, a macrophage restimulates a Th1 cell. During this interaction, the macrophage utilizes what surface proteins? Now recall that in T cell activation, there are two signals. Signal 1 is the MHC2 protein, which interacts with the TCR, T cell receptor, and the CD4, which are on the T cell. So that's signal 1. Signal 2 is the B7 protein, or CD80 or 86, that interacts with the CD28 on the T cell. That is signal 2. Both of those activate the T cell, or rather, restimulation. And that last part of the interaction is more about activation of the macrophage. And again, that's CD40 on the macrophage binding to CD40 ligand, which is on the T cell. And this leads to macrophage activation. So going back to this image, we can see signal 1, which causes T cell activation, and signal 2, both of which are needed for T cell activation. Then we can also see this CD40 interacting with the CD40 ligand, which causes macrophage activation, which basically tells the macrophage to continue doing all of its work: phagocytosis, killing the pathogen, presenting the antigens, and releasing cytokines. And that concludes this section.